Yes, so our project is the impact of chronic hypertension and antihypertensive treatment on functional hyperemia in animal model mice, this tiny white one. So our research was started from the view of small vessel diseases. As you know, the cerebral small vessel diseases refer to the pathologies affecting the microvasculature in the brain, contributing to the stroke, disability, and the cognitive decline. And hypertension, the project I talk about today, is the leading risk factor of cerebral small vessel diseases. So we worked on the functional hyperemia, which is the local increase of blood flow in active region of the brain, which contribute to the crucial to maintain the brain function, such as cognition. So we have recently demonstrated a novel signaling pathway. This capillary to arterial signaling is a major driver of this activity-dependent increase in local tissue perfusion. Capillary, the smallest vessel in the brain, senses the neuronal activity, send the vasodilatory signaling to the upstream arterioles, resulting in the vasodilation and the increase in the local cerebral blood flow. So in this study, we aimed to examine the impact of chronic hypertension on functional hyperemia, specifically on this capillary to arterial signaling using a mouse model of chronic hypertension. So we used the BPH hypertensive mice and the BPN normotensive mice in this study. And we measured the blood pressure, in vivo functional hyperemia, and ex vivo capillary to arterial signaling at 1, 4, 8 months old animals, which corresponds to approximately teens, 30s, and 50s in humans. As expected, these animals, the blood pressure was significantly higher compared to the normotensive mice and stay elevated during this experimental period. Using the laser Doppler fluorometry, we measured the functional hyperemia. As you can see here, we gave a viscous stimulation. The contralateral side of the blood flow increase. So this increase functional hyperemia, we measured as this increase as a functional hyperemia. Although the aging is another risk factor of small vessel diseases, in this model we didn't see any impairment with the up to 8 months old in normotensive mice. In contrast, in hypertensive mice, we observed age-dependent impairment of functional hyperemia in this strain of mice. Next, we examine the ex vivo capillary to arterial signaling using this newly developed preparation showing here. You can see here the red showing the smooth muscle cells or arterioles, and the green is the capillary bed. This preparation consists of isolated parenchymal arterioles, roughly 20 micrometers in diameter, and its branches transit into the capillary bed. And we stimulated only the capillary extremities with 10 millimolar potassium, mimicking neuronal activation, and measured the diameter in upstream arteriole, shown here in the blue boxes. Please see these blue boxes. So here is the example of capillary stimulation triggered arteriolar dilation or capillary to arterial vasodilatory signaling. Hope you could see here, but you can see the nice vasodilation soon after starting the capillary stimulation with potassium. So using this preparation, we examine the capillary to arterial signaling in hypertensive mice. Okay, so consistent with the in vivo observation, normotensive mice, this capillary to arterial signaling was maintained up to 8 months old. On the other hand, in hypertensive mice, we saw the gradual progressive impairment of capillary to arterial signaling. So this data suggested the chronic hypertension caused the age-dependent impairment of functional hyperemia, result mainly from the crippled this capillary to arterial signaling. Next, we elucidated whether the antihypertensive treatment alleviated the deficit in this functional hyperemia. We used 2 different antihypertensive drugs, amlodipine, the calcium channel blocker, and losartan, the angiotensin receptor 81 receptor blocker. Both drugs are widely used in antihypertensive treatment in patients, and in mice, the similarly and significantly lowered blood pressure in hypertensive mice. So again, interestingly, amlodipine, the calcium channel blocker, rescued the functional hyperemia as well as capillary to arterial signaling. However, the losartan treatment provides nearly no improvement on these outcomes. Again, the blood pressure was significantly lowered, similarly lowered in these drugs. So what's different makes this difference? So we believe the lowering the blood pressure provide a great benefit, and also conceived the losartan treatment group might have additional negative factors in these mice. So here is our ongoing study to test our novel hypothesis that the plasma aldosterone adversely impact on the functional hyperemia. It has been shown that chronic treatment of angiotensin receptor blocker, such as losartan, leads to increased plasma aldosterone levels in 30 to 40% of patients despite controlled blood pressure. So this is our first piece of the supporting evidence. We measure the plasma aldosterone levels in the ELISA kit. So this shows the plasma aldosterone levels is significantly elevated in losartan treated BPH mice. Of course, we need further studies to elucidate the role or impact of aldosterone on a functional hyperemia in animals. So here is the summary of today's presentation. We saw the viscous stimulation in this functional hyperemia is progressively impaired in hypertensive mice, likely due to the crippled capillary to arterial signaling. Antihypertensive treatment with amlodipine, but not losartan, rescued the deficit in functional hyperemia and the capillary to arterial signaling. This class effect likely resulted from elevated plasma aldosterone. Yeah, I would like to thank the people at the University of Vermont and the funding sources, and I'm happy to take any questions.